So we can start with, like, current events. So Kristen was watching the news this morning, Today Show. Apparently they were talking about that maybe your inheritance, the inheritance of your intelligence, maybe something like your IQ, shouldn't listen to the whole thing, is related to your mom somehow. So it's definitely something interesting to maybe look into and search further. My son has an autistic girl that's in his grade level, and they're doing a unit right now where they have to learn about each other kind of thing. And so he got elected to learn about autism. And I've had some experience with autism, and then we had my son was in summer camp with someone who has autism. And the thing that's difficult about that too is that there's different degrees, right? And I think you guys talking about autism as well. So I was just thinking about it too. I was like, oh, let me see if they talk about it in our book. So I did a quick search, and I was like, oh, it's in chapter 8, so we'll get to it. But you know, there's definitely more awareness out there than there used to be, which is great. And the kid that was in summer camp, he was really great about explaining to other kids, you know, why he is the way he is, and that he was okay with the way he is, and in understanding that there's something in the brain, right? So, you know, when we're talking with now 3rd graders, you know, you have to kind of talk in terms that they understand. You can't say jeans, because they're going to think you're talking about blue jeans, right? Or you have to use terminology. So there's lots of stuff out there, and so I went to a particular website, literally searched, like, how do you explain autism to kids, right? And apparently there's some game that we didn't get a chance to do because I was on my phone, and I was worried about going over my data usage when we were investigating. But the other thing that I think you guys were talking about, which I did end up seeing posted on Facebook, but I'm sure it's on some website so that I could maybe find it and share it. But I did watch the video. I didn't have a chance to listen to all of it. But the one where there was that huge square petri dish, did someone bring that up in this class, where the bacteria were mutating and being resistant to the antibiotics and growing across that petri dish? That was an awesome genetics one. Okay, so you know what? We will start out with that. And I will do something I don't normally do, because I'm pretty sure I saved it. Don't friend request me for Facebook, y'all. I won't accept. Okay. I like to talk bad about you guys on here. Just kidding. All right, where's the saved? It's different than it is on my phone. Yep. Oh, no, real life mute button. I need that one. I save a lot of food stuff, too. And teaching stuff. It should have been recent, though. So maybe we'll just search on the internet. I was hoping I had a quick saved one.
There's a really good Facebook page called Special Book by Special Kids.
Oh, I saw a reference to that. It's good?
It's very good.
Special Book by Special Kids for autism awareness.
Autism is kind of all over the spectrum of
Cool.
children with disabilities.
Awesome.
And it's like he interviews them and they talk about however they want to talk.
Let's see if it'll come up. I might have to look for it later. Was it? Where is it? Okay, I'm trying to think of keywords that are going to get me there. And of course, now they think I want to buy Petri dishes. Oh, giant dish. No. Build giant Petri. I think this is it. Yes, this is it. How cool. Okay, I'll post the link too. So, all right, so this is an enormous Petri dish. Here, I might just go ahead and play it, although I'm not sure if it's going to play. All right, let's see. So volume on.
So what we ended up building was basically a Petri dish, except that it's 2 feet by 4 feet. And the way we set it up is
with different amounts of antibiotic.
So let's start.
On the outside, there's.
It's short. It's only like.
So what we ended
4
up building
minutes.
was basically a Petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. At the base of each of these bands, we put a normal Petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, of course, thin agar that bacteria can move around in. The background is black because they're incontinent, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it 10 times as much antibiotic. And you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
How many days did he say?
11.
Frightening, huh? E. coli. Now, of course, they didn't say what antibiotic. I don't know if we read is there an article? I'm sure there is, or some thing associated with it. They would maybe tell us. But that was frightening and cool at the same time, right?
That's what freaks me out about antibiotics. Like, you don't get eaten away in that, eat medicine. People flush down the toilet and don't finish. What? Like antibiotics.
Oh, but she's talking about in food. You're talking about in food, right?
Yeah,
So there's
antibiotic resistant bacteria, say we get it. And
right.
The antibiotics don't work on us.
Right. Right. Yeah.
Pretty much.
Yeah. So no, it's not that it can, it has. We're already living in an age where there is now a bacteria that is resistant to all known antibiotics at our disposal. Okay? It happened this summer. It was discovered. Whether it happened, it was finally discovered. It probably may have existed before then, right? But they ran across a woman that had an infection, tested the bacteria against every possible antibiotic, and it was resistant to every single one.
Did she die?
I don't know.
Was it E. coli?
I don't know. I didn't keep up with it.
It was a gastrointestinal
Infection. Yeah. Yeah, it is scary. And antibiotics come from other microorganisms, right? It's their warfare that they've evolved over time, and we've just basically stolen their technology. So what my honors micro class, lab class will do, hopefully in the spring, is I'll actually, instead of doing an unknown that I know, did I tell you guys about this? They'll be taking soil samples, and it's a worldwide initiative to try and find new antibiotics, right? Because there are microorganisms out there producing antibiotics, we just haven't discovered them yet. Because they're always fighting with each other. They're always evolving, right, to outcompete. So that's the good news, the flip side of it, right? The bad news is that this is always going to happen, y'all. this is continuous, right? We know that when they copy their information, sometimes they make mistakes and sometimes they make a good mistake. They basically win the lottery because now they're resistant to something like an antibiotic, right? And it can happen very quickly. So the nice thing about this was, I think, the visual aspect for some of us too, right? To literally see, right, them changing and growing in the different concentrations. Pretty amazing. So I'll save that link for us. We can continue reading that article to find out more information if we want, or links to it. So I don't know why I'm opening another one. I should already have it open, but apparently maybe I lost internet for a minute. So I think before I started recording, somebody asked me about the PowerPoint not being posted yet. I actually kind of did that on purpose because I'm not sure how far we're going to get today. And I want to just post what we've done so that they match up with the recorded lectures. So once we're done with this one, wherever it is, I'll throw away the rest because I already have it saved as another file and post it. So we want modules, don't we? So let's post this link. 4, right? So we have one more after this one, which I think is 8. I'll look at that in a second. I don't want to. No, I'm pretty sure it's 4.
We don't have a date yet, right?
No, because it depends on when we finish. So we'll know once we get closer. Bacteria. I had it right. Ish. There we go. That looked giant. Okay, so save that for now. So let's answer that question by actually going there. So the question is, how many chapters on this first test? The plan is for test 1. Oh, it's 5. Chapter 5 we go to. But 8 we'll do later, yeah. So we have 4, which we're on, and then 5, then the test. I forget how big 5 is. I don't think we're going to cover all of 5, so in the next 2 weeks, we'll definitely probably have a test. Okay, so part 2. So now we're going to get to incomplete dominance, overdominance, and codominance. Before I get to that, remember I talked to you guys about writing out questions and I said I was going to post the thing? I'm kind of glad I didn't because I got thinking about it and I looked at your guys' results from your quiz because I hadn't looked at it before class. I think what I'm going to do is I'm going to do it as a bonus assignment for a maximum of 5 points. So it'll say 0, right? But if you read the description, it says you can get up to 5 points, which means that if you don't do it or don't get any points, it doesn't count against you. Does that make sense? And that's how I have to set it up in Canvas, right? But you can be writing your questions right now, even though I haven't posted the thing, because you can write it up in a Word document and just paste and copy it. And actually, I can put it so that you can also just attach the Word document too. So
Those points go towards that versus that.
those points will go towards homework percentage. Yes, it's a good question. They'll go towards homework percentage. Which I still need to go in there and I think I have to wait until I put a little thing in for the test before I can put in the percentages. Okay, any questions other than that? Okay. So we've kind of already gone over one of these. What one did we kind of already discuss? Anyone remember? Or know from your reading? Which one have we already talked about a little bit? Incomplete dominance, overdominance, or codominance? She's right, codominance. And what crosses were we doing when we were doing actually codominance? AB. Blood typing. Blood typing. So I'm not going to spend a lot of time on that, but we're going to go over it again, right? But that one is, I think, probably one of the easier ones. I don't know why, for us to understand. I guess because most of us are aware of blood types and the fact that you could be both A and B, right? So that makes it nice. So again, for all of these, remember, we need to be able to predict given information, right? Do Punnett squares. But also understand at the molecular level why it is we see what we see, right, for these phenotypes. So incomplete, and the definitions really for these focus on the heterozygote, because now we're not dealing with the strict, simple, dominant recessive pattern that we're used to. Instead, incomplete, as the word implies, the heterozygote is an intermediate between the two homozygous dominant versus recessive. So the example is the 4 o'clock plant. It has 2 alleles, so only 2 choices, but yet how many different phenotypes? How many different phenotypes, y'all? If the heterozygous is neither the homozygous dominant or homozygous recessive. It's its own phenotype. So now we have three. We have the wild type, red flower, right? We have the recessive, no pigment, right? A loss of function 1, you end up with a white flower. But our heterozygous, so when we mix the white with the red homozygous for each, we end up with the heterozygous, which is its own phenotype. It's pink. So remember from the beginning of this chapter, we talked about what's happening potentially at the molecular level, especially with this one, where you have only one gene that's producing the pigment, and the other one, white, means no pigment. So why are we pink? Why isn't it red?
You got some pigment, but not enough pigment.
Right, you got some pigment, but not enough. And we know lighter shades of red appear what? Pink. So one of the possibilities is that we only produce half red, right? Which ends up being pink. Does that make sense to everybody? So one is it that you just produce half, right? Because you only have one gene to read. What's another thing that could happen, which is why sometimes we don't end up with this intermediate? Even though you're a heterozygous, right? And you got maybe a defective gene from one parent and a good gene from the other parent, what does your body do? But you need this, right, to function. So could you be okay with just 50% of a protein? Yeah, you could have the normal phenotype and we don't notice unless we really look at it at a molecular level, that you're only producing half as everyone else. Or your body is upregulating, right? Producing more of it from just that one copy than it would if it had 2. Upregulating. Make sense? So it's either where 50 is enough, right? Or you produce more than 50. You upregulate. Here, 50 is not enough to be red, right? They don't upregulate, so they end up being pink. Make sense? Everyone still with me? Yeah, maybe so. Okay, so what they do here is they denote C for color, and the reason why they're doing 2 letters too is to denote this different characteristic, right, that we're dealing with incomplete dominance. So they do the R for dominant and W, which gets kind of confusing, right, because then you're like, okay, what does that mean? Are you mixing these two? No, it really means that we're only just producing half red. Does that make sense to you guys? I don't like the way they denote this, quite honestly. I find it confusing, right? So as we said, it's not sufficient, so you don't end up, and so we get a little bit different ratio here, right? When we cross the heterozygous individuals, instead of the usual 3 to 1 that we're used to, right, we end up with a 1 to 2 to 1 ratio, right, where you got 1 red, 2 pink, and 1 white. So given this problem for the 4 o'clock, what is the expected outcome if we cross a pink flower with a red flower? Okay, so I'm going to change up the way we write this, right, genotypically, because I find it makes a little bit more sense for me. Okay, so, oops, I forgot I have to be in the other mode to be able to write. And the answer's there. Shrink that. Did it a different way. Okay, so I'm just going to write color, right? And where the heterozygous we know is pink, right? And the homozygous dominant is red, right? And the recessive is what? Homozygous recessive, white, right? So I'm going to do, maybe, I must have left something open. I don't want that dinging the whole time. Yep. That'll make noises. Let's turn off all that extra stuff we don't need. Okay, if I could click on the right thing would be good. Okay. Now it's wanting me to do something else. Okay. Stop. Oh, did I mess up my thing when I imported it in? Yep, I did. Oh, that bites. Do I still have it open? Yes. Okay. Here, let's just steal, like, this one. Or, yeah, this one. Copy. Voila. Okay, so I'm going to change it to C's, big C, little c. I think that's the other thing is like C is hard to see. Let's do the R's. Let's just say R, right? R is easy to see. So, but one's going to be heterozygous and one's going to be homozygous, right? So I need to change this to RR, and this is red, right? That's what we're saying. And this would be also red.
Can we do it with the CR and CRC or CR and CW to make see it better?
You think it makes better sense that way? Yeah, we can write it both ways. Pink. You see? I find that's easier, right? Because we know that the homozygous red has to be 2 dominant R's, right? And then the heterozygous, capital R, little r, we know is not red, it's pink, right? So we end up with half red and half pink. But I'll honor your request, and I will fight through oops, it's going to be a little bit off the page. Come up here. All right, so we would write this CR. I don't know if I can do that.
Make one C capitalized in your lowercase.
No, it's a superscript. It's part of the reason why I don't want to do it on the computer. Where is it? Font super. But once I do it once, I can just copy it. Right, and then the next one would be W, right? So it makes it hard to see. So then phenotypes are going to be the same. I don't understand why they do that W, it confuses me. So they're not all that different. The important thing is it doesn't matter how you denote it, just as you know what your symbols mean, right? Does that make sense to you guys? So I shorthand it a lot of times. I mean, I could even, you know, make these W's, say, write it, right, to keep it consistent, and just get rid of the C. The important thing is you know what the symbols mean, right? Everyone still with me? So like you'll notice too when we get to the blood typing, I never wrote the HB, right? I just wrote the A or the, I just wrote the superscript. And bother writing out the other half. You don't really need it. I mean, and the reasoning for it is, right, so that when other people look at it, it's a way of designating that there's a different type of dominance than we're used to, right? It isn't straight up dominant recessive inheritance. Does that make sense to you guys? Okay. All right. So the answer is D, right? There we go. So now it's not hidden. So another example of incomplete, when we look at it a little closer. So actually knowing whether something is truly dominant or incomplete dominance, you'd have to examine it a little bit closer sometimes. So it just really depends on the level. So for example, the characteristic for the pea plant, for the shape of the actual pea. So we thought that the heterozygous, that one gives round, right? And it really does. Phenotypically, when you look at it, it's round. And this is a case, again, where 50% is enough to make it look round. So if you just look at those two, they look round, right? And this guy is definitely wrinkled. But if you were to look at them microscopically, you would find this one produces more of the proteins that support that round structure, where this guy doesn't produce as many, but produces enough that it still ends up being round. Where there's significantly less of that protein produced for the wrinkled ones, right? So they can't hold that round shape in there, wrinkled. So it looks, right, when we just look at them phenotypically, right, it's like, oh, it's a strict dominant recessive relationship. But in truth, if you look at it at a microscopic level, it's incomplete. Depends on what level you're looking at it, right? Because there's truly, there's 3 different phenotypes we're seeing here, right? Everyone agree with that? All right. So he's definitely an intermediate. The next is OverDominance, and most people know the classic example for OverDominance, or one form of OverDominance, and that's sickle cell anemia. And we've kind of already a little bit touched on this at the beginning of our course. So this is a phenomenon in which the heterozygous is better suited for the environment than either of the homozygous, right? That's what I mean by more vigorous, right? You're the fittest out of the two. So this is also sometimes referred to as the heterozygote advantage, right? Because they're better. So the classic example is sickle cell. It's an autosomal recessive disorder, which means it's not found on your sex chromosomes, all the other. It's found on another chromosome. I don't know what number, though. We need to find out so we can find it. I think we should start, like, highlighting on my poster which ones we talk about, which diseases, and keep track. So we already did Huntington's, right, which is on 4. We need to find sickle cell. So, and cystic fibrosis too, right, because we've talked about that. So this is where affected individuals produce an abnormal form of hemoglobin, right? So the hemoglobin isn't the normal form, it's mutated. So there's just 2 alleles, right? And again, it's one of those where they write HB for hemoglobin A, which encodes for the normal one, and then S to denote for that it codes for the abnormal hemoglobin, which can contribute to having sickle cell.
Can you test for sickle cells while they're still in the womb, or is that something we should figure out later?
I don't know. But that's definitely like something like if a parent had sickle cell, hopefully they would be talking with their doctor about, you know, what are the chances if they had kids and about when testing would happen. So if somebody has both copies of deformed hemoglobin, then they have the disease we know as sickle cell. And so under low oxygen conditions, this causes a deformity of the cell, such that it looks like a little half-moon sickle shape. The 2 major problems with this shape is not the normal shape, right? So it greatly reduces the lifespan of the red blood cells, this abnormal shape. And so you got to work harder to produce more red blood cells more often. And so typically people tend to be anemic. The odd shape also clumps up much easier than the normal shape. And so this can partially block capillaries and affect circulation. And thus, because of these complications, people with this disease typically have a shorter than normal lifespan. There's a picture, right? Our normal biconcaved, right, increased surface area for gas exchange, and one that is sickle shaped. So it's in a really high frequency in African Americans where malaria is found. So we ask why? Well, why is that? Why would something that is detrimental, shortens your lifespan, why would that gene be more common, right, to be sickle cell, to have that gene be more common in the population? And it's because the protozoa Plasmodium, and there's 4 different species that causes this disease we know as malaria, infect the red blood cells in part of their life cycle. There's 2 parts of their life cycle. one part they infect the liver cells, but they also infect and multiply in the red blood cells. So inside the mosquito, too, it goes through part of its life cycle, the sexual part of its life cycle. Other part is in the red blood cells. So people that are heterozygous, which means that they produce some hemoglobin that's defective and some good hemoglobin, because you make so much hemoglobin, you use both copies that both parents gave you. So the bad news here is that some of your copies are not good. The good news, though, if you're heterozygous, where you have some bad copies and some good copies, having that deformed hemoglobin, when the plasmodium infects, the cell ruptures. And so they're not able to multiply inside that red blood cell. So it stops a portion of the life cycle of them replicating within the red blood cells. Does that make sense to you guys? So it's one of those, you know, kind of fortunate things. So therefore, if you are heterozygous, this ends up being an advantage, right? But what do we know if two heterozygous individuals mate? What's the chances that a child is going to end up with both sickle cell genes? No, two heterozygous individuals. 25%, 1/4. Right? But again, those are going to be sick with sickle cell, right? So most likely those people are not having children for the most part, especially in Africa where they don't have medical treatment to the level that we have maybe here, or access to it. The people who are homozygous, they don't have this advantage, right? So they get malaria and they die from malaria and they don't have kids. And instead, the people who are heterozygous, they survive malaria, right? They don't have sickle cell, full-blown sickle cell, right? They produce enough hemoglobin to get by. They mate and have kids, but then, boom, you end up with kids with sickle cell, right? You end up with some that are heterozygous like the parents, about 50%. Right? And then you end up with ones that don't have the sickle cell trait, but probably will, because of the environment, get malaria. So you see how the heterozygous ends up being a large number in that population because it is beneficial. Because of that selective pressure of that environment of the plasmodium that causes malaria. Still with me? All right. Mm-hmm. They have a 25% chance of getting it. Do we do this cross? I can't remember. No, but we will. I think we do. We'll get to it. All right, so the next one. So at the molecular level, it's due to two alleles that produce slightly different proteins, right? So each copy of the DNA is copied, right, and we make proteins from it. So how is this favorable, though, right? One is the example of the disease resistance, right? So because you're heterozygous, you are protected from this disease, right? So that's beneficial. The problem is when two heterozygous people mate, right, then you have the possibility of someone having sickle cell, right, or being more susceptible to the disease, malaria. The other is the Homodimer Formation, which we're going to talk about next. Or variation in functional activity, right? How they function differently, right? The proteins function at a different activity level. So the disease resistance example, right? So say you produce two good copies, two normal copies of hemoglobin, right? You got two good copies from both parents. The bad news is this pathogen is going to attack your red blood cells, multiply in them, and bust out and attack more red blood cells and keep doing this over and over again, right? you're going to have malaria, which is pretty horrible. If you're the heterozygotes, right, you have one normal, but then you have one abnormal gene, right? So because of that, you're resistant to this particular pathogen. Slightly reduced. So therefore, they have slightly reduced protein function. So, right, the heterozygous, right? But again, even though it's reduced, it's not reduced to such a level that it's significant that you would say someone has sickle cell, right? Has that disease. It's not enough to actually cause the really serious side effects. But enough to prevent the disease, which is great. Right? Malaria, anyways. So, sickle cell anemia, right? That link of that defect with malaria is an example of this. Tay-Sachs disease and tuberculosis resistance, believe it or not. And I haven't looked up the specifics with this, but I know, like with tuberculosis, it can grow inside our white blood cells, our macrophages. And so I'm thinking it's probably one of these things too where it like doesn't get in because of the deformities that happens in Tay-Sachs, but I don't know for sure. We need to look this one up. And then PKU and fungal toxins. And so PKU, remember, is not having the enzyme to convert phenylalanine into tyrosine, not having the enzyme that does that, so they restrict phenylalanine in their diet. And so that would make sense too. There are some organisms that really need certain amino acids in their environment. And so probably phenylalanine is one that's like needed in high concentrations for fungi. And so the fact that these people don't eat it very much, it wouldn't be readily available to the fungi. And so therefore it would stop their ability to grow. Again, I'm educatingly guessing on these. Right? You might want to look them up and see what it actually is. See how close I am to the truth. The next is Homodimer Formation. So this is where you actually, dimer means what? Two, right? So you produce two proteins, they come together to form that function. And in fact, hemoglobin, I want to say, is might even be a pentamer. I think it might be 4 proteins that come together to form the actual hemoglobin molecule. That's the one they like always make all the time, like when they do protein classes. Trying to find. Yeah. Look, see, it's 4. Ha. Two A's and 2 B's. So homo meaning the same, dimer, right? So they I think make 2 copies of A and 2 copies of B, and then all 4 of those come together to form the hemoglobin molecule. And then you have the heme group in there that complexes with the iron that actually interacts with the oxygen. Pretty impressive molecule. Protein people love hemoglobin. It's like the gold standard of proteins. There's so much known about it, everybody talks about it. All right, so homodimers, so composed of 2 different subunits, coded by 2 alleles of the same gene. So you could be AA1, and you make only those copies. Or you could be the other choice, the other allele, right, A2. Or you could be heterozygous, right? You could have the number 1 form and the number 2 form. Because of the information that you have, so they get made separately, right? So you make A1 or A2, and then they combine with each other. But since this person makes A1 and A2, when they combine, sometimes 2 A1s will combine, or 2 A2s, or an A1 and an A2. You have 3 different phenotypes, genotypes, or no, phenotypes because they're proteins.
Is A1A2 a homodimer or a hetero?
No, this is homo, right? So they're homodimer means that,
The A is the same?
yeah, the A is the same. So that component is the same. The A is the same, right? The proteins are similar. They interact with each other, right? They'll still bind with each other. But in the case of this individual, it's a homodimer in that the A's are the same, right? But the proteins are slightly different. 1 and 2 are slightly different from each other. Still with me, Angela? Are you with us? You mulling it over? When they're saying homodimer, they're referring to the proteins, 2 proteins that are similar coming together.
Right.
The 2 A's.
Like, could you also, if you're using just the letters, would it be kind of like A1, A2 in one column, and A1, A2 in the other, whereas the heterozygous is A1, A2?
Right, so look at this picture. So these are 3 possibilities of proteins that this person could make because genetically they have both the 1 type and the 2 type. They're heterozygous. Yes, so, maybe so. Okay. Maybe it'll sink in. And come to find out, right, when you put 1 and 2 together, they act better. They function better than the 2 1s together or the 2 2s. So this is where activity level matters, right? I mean, no. I don't know. They call this homodimer function. Formation. 2 things together. All right. The next is variation in functional activity. This one's much easier to understand. So you have an enzyme for some metabolic pathway, right? Say splitting glucose into 2 pyruvates, right? One of the enzymes in glycolysis. E1, that particular enzyme, that variant, that allele, right, if you've got that, you can work within the optimum range of 27 to 32 degrees Celsius. So at low temperatures, right, you work really fast. That's your optimum range, right? You're like Speedy Gonzalez breaking apart those glucoses. But, you know, when it gets too warm, right, you're not going to do it as effectively, or too cold. The second guy works in the higher ranges. So, if you're a heterozygote, what does that mean? Which one of these do you produce? Both. Heterozygous means different, right? You have one of each. One from one parent, one from the other. So you produce both. So why would that be beneficial? Because you're going to work really fast if you only have E1 just in this range, right? So you're limited, right? If you have only E2, then you work in the higher range. And you're not going to work as well outside of those ranges. But if you're a heterozygote, you produce both these enzymes, so it doesn't matter whether it's high or low, you're still going like gangbusters, right? You're going fast. So, you know, I carry an umbrella and a rain jacket. Because if it's windy, the umbrella goes away. Just stick with the rain jacket. Double coverage.
I'm not really seeing a big difference between the Homodimer formation and its functional activity. So are they basically the same mechanism? It's just a different example?
No, yeah, I know.
Or is the Homodimer a subset of functional activity? Does that make sense?
Yeah, I mean, they put it as two separate things, but there is definitely some relationship between the two in the examples that they're giving. And they do not, for this one, give us any specific examples. That's why I kind of said, you know, imagine an enzyme in glycolysis, right, to help us conceptualize it a little bit better. So I'm going to look for next time for us for some like real world examples of these that we can kind of wrap our heads around a little bit better to distinguish them, or realize that really there isn't much distinguished between one. The only thing I think of is this is a single enzyme, single protein working on its own, right, where the individuals are producing both, where the difference here, so they seem really similar, but the difference here is it's actually two proteins coming together. You got it, Aaron? So the distinctive difference they're making here is that it's two proteins working together, homodimers, two together.
The other one's just one.
Where and in this case, then also, right, the benefit of this is that the two that are different work better together than either of the respective homozygous. Still with me? So, as we said, it's better when there's two proteins, right? This is a homodimer formation. It's better to have two different ones than two of the same. Where here, we're not talking about two proteins coming together. We're talking about single proteins, singly produced. And the fact that if you're heterozygous, you produce both of them, but each one of these are slightly different, right? They still do the same job, right? They both say do glycolysis. But the difference is their optimum ranges, right? So these proteins are slightly different from each other in their activity level based on temperature. So you're better off if you've got both, because then you can adjust to different levels of temperature, right? Very easily. Where somebody who only produces this enzyme, if it gets too warm, right, they're not going to be as efficient at breaking down glucose, say. Where somebody who has this one is stuck in this range, where if you're heterozygous, you have both, so it doesn't matter temperature, right? Is that better? Okay. I am still going to try and find us some real life examples. Okay, so multiple alleles. So this is when we can get into what? So we've talked about incomplete dominance, overdominance, what's left? Codominance. And you got to have more than just two alleles for this. So within the population, there's more than just two choices. In the case of ABO blood group, there are three. All right, but there could be more than three. But if we're talking about diploid individuals, right, a person, you can only have two copies. right? Diploid means 2 copies. And then, of course, your sex cells only have 1 copy, right? They're haploid. Everyone still with me? So although within the population, right, for us, for ABO blood group, there are 3 potential alleles out there that you could have, right? You could have A, you could have B, or you could have O, which doesn't produce A or B. But as an individual, you can only have 2 of those 3, right? Because you're diploid. Yes? Heads go up and down, please. Yes, okay. And then when you make your sex cells, you're only going to give one of those 2 that you have to your children. And this is why, to a certain extent, blood typing could be used for paternity, right? Because if you say, and you and your spouse are both A, right? And then all of a sudden your child is B. That is not actually your biological child, is it? Unless you're some type of chimera or something, right? If it's a normal thing, nothing wonky happened, where did that B come from? Not the 2 A people, right? Because then their blood type would be AB or B. Okay. So you produce molecules on the surface we commonly refer to as antigens because antibodies will bind to them, right? Immune systems will attack ones they do not recognize as self. So again, they do the whole superscript, up, Erica, up, right? Where you produce the antigen that we know as A, your A blood type, or B, or little I for no antigen. So they have this component, N-acetylglucosamine, is added. So see, we already have, I think, what are these, sugars? Yeah, they're sugars. So we have galactose and N-acetylglucosamine and a fructose. You know, old people like me, we do have these sugars on our surface of our red blood cells, but they're not antigenic. Like antibodies don't bind to these, right? They're not different enough, I guess. For the A, though, they add an extra molecule. They add the N-acetylglucosamine. And for the B's, they add an additional galactose. And that makes this antigenic. And then, of course, ABs produce both on the surface of their red blood cells, both A and B. Right? What we refer to as the A and B antigen. So right down to the molecular level, they know what these antigens are made of and what the differences are, which is pretty impressive. So I is recessive to both A and B. A and B together are both expressed, so they're said to be codominant in the heterozygote. Yeah, so this just talks about the sugars. So a fourth can be added by an enzyme. So, as I said, there was that core 3 that we looked at, right, in this picture. Core 3, 1, 2, 3, right, this one's repeated. But they'll add a fourth one, depending on whether they're A, which codes for the N-acetyl, or B, for another galactose. I'm not going to ask you in this level of detail on a test, right? But we know this, right? And so they're recognized by different antibodies, right? We'll be able to bind to these different forms. So a particular enzyme actually adds it on there. So for safe blood transfusions to occur, the donor must The donor's blood must be an appropriate match for the recipient. And the easiest way, y'all, to remember this is you never want to give somebody antigens they don't have, right? Because then their body's going to go, that's not me, attack, right? You need it to look as similar to them or be invisible, like O is, right? Antibodies don't attack O. So, for example, if type O individual received blood, right, from anybody with A's or B's or A and B, that would be really bad, right? For me, I can only get O. You can't give me those other antigens, right? I have antibodies that will attack A and B. Right? It'll agglutinate, it'll be a big old mess. So the example they give is a person with type B, and I know that this Punnett square is messed up too because I put it in there the same way I put the other one in. So I gotta steal. Let's see, what is it? Because I already did out some of these, huh? I'm just gonna take one. Take an easy one and modify. Angela, you gonna let me do it shorthand? I'm not gonna put the I's. I'm gonna use A, B, and O. Okay, so we say an AB crossed with an O, right? And O would be little I, right? But I'm gonna write it O, O. Just delete these. So we end up with AO, which is still what blood type? A. Right? As long as you have that one dominant, right? You're going to reduce that A antigen. Then we're going to have BOs, which is also type B. And another AO, right? And another BO. So what are the possibilities of blood types for the children? Just A and B only. Yeah. No O's. And I think that's it for today. So be working on your questions. And if I had guessed, you were going to get to this far. Pretty good at pacing myself. I should have guessed. And then continue reading in this chapter, right, so that you have an idea what we're talking about. And I love sharing media stories and genetic stories and stuff like that, so, you know, feel free if you find something interesting. Even email it to me ahead of time so I can research it ahead of time.